This is an overview of photosynthesis. It's the capturing and converting of light energy from the sun in the chloroplast through light-independent reactions. Photosynthesis starts when light energy enters the thylakoid membrane. The light-dependent stage of photosynthesis happens in the thylakoid membrane. It's when photons of light are absorbed by chlorophyll pigments of photosystem II. The absorption of this light excites electrons within the chlorophyll molecules. The excited electrons are now taken up by an electron acceptor. They are then passed through a chain of electron carriers and will end up at photosystem I. This proton pump uses the energy from the electron transport chain to pump the excess hydrogen ions into the thylakoid space. The movement of the hydrogen ions going down the concentration gradient is called chemiosmosis, how it goes from high to low concentration. This is what drives ATP production. The ATP that is produced will be used in the Calvin cycle. Light energy also enters photosystem I. The electrons are re-excited and passed up the electron acceptor. The electrons then pass down the electron transport chain. While this happens, NADP+ reduces to NADPH, which is used in the Calvin-Benson cycle. The Calvin-Benson cycle, our light-independent reaction, has 3 stages, 1, 2, 3. The first being carbon fixation. It starts with RUBP, which stands for ribulose biphosphate, a 5-carbon compound that is added to the input of 1 carbon, which gives us an unstable 6-carbon compound. Because it's unstable, it immediately breaks down into 2 identical 3-carbon compounds. These are our first stable products of the process. Our second stage is the reduction stage, which starts with the 3-carbon compounds, but they are in a low energy state. So, they must be activated by ATP, which we can see here, as well as be reduced by NADPH. The result of this reaction is a molecule of PGAL, which is the output of the reduction stage. Glycolysis starts with glucose, a 6-carbon compound. 2 molecules of ATP are used to start the oxidation of glucose. 4 molecules of ATP are produced and 2 NAD are reduced. This splits glucose into 2 pyruvate molecules. Each pyruvate then removes CO2. NAD is then reduced to 2 NADH, which produces acetyl-CoA. The Krebs cycle. It starts when acetyl-CoA, a 2-carbon compound, is picked up by a 4-carbon compound, which results in a 6-carbon compound. NAD+ is then reduced to NADH, and then 1 CO2 is removed as a waste product, which results in a newly formed 5-carbon compound. Another NAD+ is reduced to NADPH, as well as another CO2 is removed, producing a 4-carbon compound. An ATP is produced by the reduction of ADP plus P, as well as FAD is reduced to FADH. Our last reduction is another NAD plus reduced to NADH. We are now back at our 4-carbon compound since acetyl-CoA has been regenerated and is ready to pick up more carbon groups. The last stage is the electron transport chain, which occurs in the inner membrane of the mitochondria. Electrons from FADH and NADH pass on to electron carriers in the membrane. As you can see, the yellow arrow is pointing to the carriers in the membrane. As the electrons are being passed, energy is released in small amounts, and the energy is used to pump hydrogen ions across the membrane from the matrix to the intermembrane space. It moves into the intermembrane space by the ATP synthase. This allows hydrogen ions to move from a high concentration gradient to a low concentration gradient. This is called chemiosmosis. as this is also where vast majority of ATP molecules are produced, as well as stored. We now move on to anaerobic respiration, which is respiration without oxygen. This type of respiration happens in animals, particularly in the muscle. It starts off with glucose, which can still go through glycolysis since it does not require oxygen, where 2-NADP is reduced to 2-ATP, and where 2-NAD is reduced to 2-NADH. This produces 2 pyruvate molecules like before, but in this case, then reduces further into 2-NAD+. This produces lactic acid. We will now be talking about fermentation, which happens in plants. It still does not require oxygen, but it is different from the other types of respiration because its end product isn't pyruvate or lactic acid. It again starts with glucose and glycolysis. We start with 2-NAD and 2-AP plus P, and they reduce to 2-ATP and 2-NADH. This still produces 2-pyruvate, yet later gives off CO2, and 2-NADH is reduced to 2-NAD+, which leaves us with our final product of ethanol.